Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the immunoglobulin A. So, from the last couple of videos, we are talking about the isotypes of the antibodies, and we talked about the immunoglobulin G and its four subtypes in detail. In the last video, I've told you about the immunoglobulin A and one of its specific type, which is known as the monomeric immunoglobulin A. Now, in this particular video, I want to focus on the dimeric immunoglobulin A. What I mean by that is that in contrast to the immunoglobulin G, which is usually present in the monomeric form, you can actually see the dimeric form of the immunoglobulin A as well. Now, when you talk about these immunoglobulin A, they are actually making up about 15% of all immunoglobulins in the human body. So, this is the second most common when you talk about the concentration or their abundance in the body. So, immunoglobulin A is making the second most abundant immunoglobulin of all immunoglobulins in the body, and it makes about 15% of the immunoglobulins. Now, immunoglobulin A exists in both the monomeric and the dimeric form. In the last video, we had detailed discussion on the monomeric one. In this particular video, I want to focus on the dimeric forms. Now, where are these monomeric and dimeric immunoglobulin A found? When you talk about the monomeric immunoglobulin A, they are actually found in the internal fluids like the plasma and in the cerebrospinal fluid. Now, when you talk about the dimeric immunoglobulin A, they are actually found in the secretions. For example, you can find them in the saliva, the milk, the colostrum, tears, and secretions from the genitourinary tract, the gastrointestinal tract, and the respiratory epithelium. So their location that is different. They are found in different parts of the body. So the monomeric and dimeric immunoglobulin A, you can expect them in different parts of the human body, for example. Now let us talk about the structure of the dimeric immunoglobulin A. We had detailed discussion on the monomeric immunoglobulin A, and the dimeric would mean you are actually combining two monomeric immunoglobulin A to each other. Now this is the structure of the dimeric immunoglobulin A. If you look at this one is the light chain. This one is another light chain. This is the heavy chain starting from here, moving up to here. This is another heavy chain starting from here, moving up to here. So this is one antibody. So by dark pink, I'm representing by these heavy chains, like these one. These are actually the heavy chains, and these one. They are actually the heavy chains over here. Now the light chains, I'm representing them by blue color. So this is light chain, this one is light chain, this is the light chain, and this one is the light chain. Now, the special thing about the dimeric immunoglobulin A is the joining chain, or which is known as the G chain. And you can see this dark purple over here, which is actually wrapping around these two monomeric immunoglobulin A. This is known as the joining chain or the J chain. Before going into the J chain, let us see how there are two monomeric immunoglobulin A in the dimeric immunoglobulin A. So we are going to focus on the monomeric first. So this is the monomeric starting from here, moving up to here. This is one antibody starting from here, moving up to here. This is another antibody. So this is the light chain. This one is the light chain. So two light chains starting from here up to here. This is one heavy chain starting from here, moving up to here. This is another heavy chain. So this is a one monomeric immunoglobulin A having two heavy chains and two light chains. Similarly, over here, you can see this is one light chain. This is another light chain starting from here, moving up to here, one heavy chain. Starting from here, moving up to here, this is another heavy chain. So two light chains, two heavy chains. So this is actually making another monomeric immunoglobulin A. So you're actually joining two of them. Therefore, we call them as the dimeric immunoglobulin A. But there should be some mechanism by which you can join these two monomeric immunoglobulin A to each other. And this is actually the J chain or the joining chain, which is responsible for dimerization of the monomeric immunoglobulin A. Now, this G chain is actually a polypeptide chain. made up of about 137 amino acid, and it has a very high content of the cysteine residues, like there are 8 cysteine residues in the G chain. So why there is a very high content of the cysteine residues over here? This is because that this cysteine residue, they are very crucial for forming the disulfide bonds, and these disulfide bonds, they are actually linking the J chain to the Fc region of the immunoglobulin A antibodies, allowing for dimerization. Look at it here. This one is the Fc region. So this would be the Fc region of this antibody. This is going to be the Fc region of this another antibody. Now, what the J chain do is it is actually joining these two Fc regions to each other, and these are the Fc regions. And when they are joined to each other, they are actually joined through the formation of the disulfide bonds. And the cysteines for these sulfide bonds, they are going to be provided by the J chains on one side, and of course by the Fc regions on the other side. So the J chain, that is very important, that is very crucial for forming the disulfide bonds, thereby converting the monomeric immunoglobulin A into dimeric immunoglobulin A. Now you might be seeing another component over here, this one, this secretory component over here. Now the secretory component, that is, you can say, performing two important functions. The first function is they are responsible for secretions. As I've told you, that the dimeric immunoglobulin A is usually found in the secretions like the tear, the saliva, or the colostrum, for example. So how they are secreted? The secretory component is actually responsible for this secretion. Secondly, the secretory component is very important to protect the dimeric immunoglobulin A from being degraded by the proteolytic enzyme. For example, if you talk about the gastrointestinal tract, a lot of proteases, they are there. So what they are going to do is they have the ability to break down the protein like the immunoglobulin. So the secretory component is actually preventing the proteolytic degradation by these enzymes, and hence the secretory immunoglobulin A or the dimeric immunoglobulin A, they are protected from each other. Don't worry about this. If you talk about this secretory component in the coming video, because that is very important. So how the secretory component is produced, how it works, that would be the focus of our next video. But just in this particular video, keep in mind that the secretory component is for secretion and protecting the dimeric immunoglobulin A. So this is a comparison. This is the immunoglobulin A monomer, one light chain, another light chain, one heavy chain, another heavy chain. You've got two light chains and you've got two heavy chains, a monomer. If you talk about the dimeric one, so you are going to see 1, 2, 3, and 4. 4 heavy chains, 4 light chains, starting from here up to here. One heavy chain starting from here up to here. The second heavy chains, this is the third heavy chain, and this one is the fourth heavy chain. So in the dimeric one, you are going to see 4 light chains, and you are going to see 4 heavy chains and a J chain over here. When you talk about the secretory one, so that would be the dimeric immunoglobulin A plus the secretory component. So that is going to make the secretory immunoglobulin A. So these are actually the 3 different forms of the immunoglobulin A, and you should keep that in mind. And in the next video, we are going to have a detailed discussion on the secretory component and how that is responsible for the secretion of these dimeric immunoglobulin A.